as a welcome everybody. We'll get started at 5 after the hour. So if you want to go grab something to drink or need extra time to get ready, don't panic. We won't start without you. We'll get started in about 1 minute. All right, welcome everybody. Exciting event, exciting week for the microbiome. Today's roundtable discussion will all be about the microbiome and unlocking the biorevolution. Rules of engagement, put your questions and or comments in the chat or Q&A box. The Q&A box is set up so that if you had a particular question for one of the panelists or panelist companies, etc., they could go in there and answer your question. Maybe it'll get promoted to a conversation, maybe it won't, but at least you get your question answered. Maybe you have to run to a different meeting or somewhere else. This way you ensure that your questions get answered by the panelists today. So that's an important point as well. We're going to do our best to moderate questions with the panelists. We'll have questions for the panelists. The panelists will have questions. So it should be very interactive. Don't hold back. Obviously, don't ask proprietary questions or things like that because they're not going to get answered. But let's really have a great discussion today about the microbiome and the revolution that's happening in front of us. This will be recorded and go on demand. So again, if you have to leave for some reason, no worries, you can go back and watch it. So today's agenda, panelist introductions, brief industry perspective, roundtable discussions. So today's panelists are Barry Murphy. He's the microbiomics science lead at Unilever. Jeanette Jones is the director of integrated biology R&D at Unilever. Anthony Finbow, CEO, Eagle Genomics. Dana Walsh, a hands-on microbiome scientist. Arlene Fosmer, Innovation Director for Cargill Health Technologies. Mike Romanos, CEO of Microbiotica, and Alex Mitchell, Director of Bioinformatics at Eagle Genomics. So let's just kind of start with the industry overview / perspective. I think most people on this call know, but if you don't, the whole microbiome movement is massive. So don't underestimate the complexity and what this is going to bring to human understanding, for years to come. So we're going to touch on some definitions as they maybe stand today, maybe some interesting facts to pique people's interests, maybe to create controversy. And someone on the call will say, that's not true, or I know more about that, or this or that. So it could be end up part of a discussion. Maybe some cause and effects that we see. And then lastly, and extremely importantly, the scientific informatics and scientific data management guidance that comes with all of this. So let's just start with some definitions. Microbiota, the assemblage of microorganisms present in a defined environment. The controversial one, microbiome, the entire habitat. This is a condensed one. We'll get into this a little more in a second. The entire habitat, including the microorganisms, bacteria, archaea, lower and higher eukaryotes, and viruses, their genomes, and the surrounding environmental conditions. Pretty encompassing. Metagenome, the collection of genomes and genes from the members of microbiota. Metataxonomics, the high throughput process used to characterize the entire microbiota and create a taxonomic tree showing the relationship between all sequences obtained. Precision microbiomics, the use of the gut microbiome as a biomarker to predict the effect of specific dietary components on host health, and the use of these data to design precision diets and interventions that ensure optimal health. And then obviously popular terms as it comes more to the consumer, probiotics, live microorganisms intended to have health benefits when consumed or applied to the body. Prebiotics, nutrients degraded by gut microbiota that promote the growth of beneficial microorganisms. Postbiotics, substances released by microorganisms known to exert a beneficial effect on the host. Symbiotics, a mixture comprising live microorganisms as substrates selectively utilized by host microorganisms that confer health benefit on the host. So what you're kind of seeing here is a systems biology that exists today, a highly complex one that we're just starting to understand. And you've probably heard this many times in your career or your academic career, is that we really don't know much about biology today. We're learning and we're making discoveries, but 20 years from now will be an exponential understanding compared to today. And it's because of microbiome research, multiomics, et cetera, that we're getting into details and we're starting to put away biases and learn and understand more. And what comes along with that is, oh my gosh, we're gonna have to manage all this data and analysis, and we're gonna have to do it right the first time. So a clearer consensus definition of the microbiome among researchers from diverse fields remains debatable, and this is true. And in some cases, there are people who say absolutely not. There's this definition, and that's what we're going with. But just to run through this, and I just put it in slides if people want these slides to share later, and it's from the paper from Gabriel Berg. But there are ecological definitions that for some feel that it's good enough. There are organisms host dependent definitions. And then there's genomic method-driven definitions. And then some folks have combined these. And then what you'll see is we actually feel there's, and others do feel there's a better definition. So we have ecological definitions on, they describe the microbiome following concepts derived from ecology or multicellular organisms, right? But it probably doesn't go deep enough. and the reason it doesn't go deep enough is because you need to uniquely identify a lot of these things in order to work with them, share, collaborate. Otherwise, after a period of time, it becomes very difficult to really understand what that entity was, and it wasn't well characterized enough. There's this host or organism dependent definition, and they're based on microbial interactions with the host. But again, there's the complexity here is much more than that. So being able to get down at the genomic level, the metagenomic level, and understand and classify is critical. Then you have the strict genomic method. But as we all know, there's epigenetics, there's all types of things, there's systems that go on. So understanding at a genomic level is fine, but again, it's part of the picture. And actually, it's a big learning for humans just from a research perspective. There's been a trend over the past 15, 20 years to go all genomics. And why we're here talking about this today is many others have realized that it's not just about the genomics. There's so much more going on, and physical and biophysical interactions going on that we need to do a better job to describe. So then people said, oh, well, let's just combine these definitions and we'll get where we want. But again, that kind of fell short and it was incomplete, like the picture. So the definition that many in the industry have hung their hat on was an original definition from 1988 from WIPS, but another group of researchers actually put a fine point on this and say, the microbiome is defined as a characteristic microbial community occupying a reasonable, well-defined habitat, which has distinct physiochemical properties. It not only refers to the microorganisms involved, but it also encompasses their theater of activity, which results in the formation of specific ecological niches. The microbiome, which forms a dynamic and interactive microecosystem prone to change in time and scale, is integrated in macroecosystems, including eukaryotic hosts, and here crucial for the functioning and health. So this is what today, what we're saying encompasses the microbiome. And it's up for debate, of course, and we can talk about some of this today, but it seems to be a very holistic approach to looking at this. And why is that? Because in the world that we live in today with high performance computing, machine learning, artificial intelligence, the massive amounts of data that are going to be coming through will not be able to be analyzed by humans alone. We will need that computer assistance. And this is why having this definition formed out early and it's accurate is very important. So some interesting facts. Since the 3rd century AD, the microbiome has been studied, assumed, talked about, etc. And it's been a long time. Over the period from then until now, there's been some discoveries, there's been discussions, but it hasn't really caught on like it's caught on today. And so that's another point of topic today. It's estimated there's 4 times 10 to the 13 microbial cells in the human body, which is matching our human cells. But the microbial genes outnumber the human genes by 500-fold. Again, some of these numbers are up for debate. The human microbiota impacts the physiological of the human body in different ways, acting as a mechanism of defense, skin and gut, processing and eliminating toxic substances or indigestible food components, generating influential metabolites, beneficial or toxic, and in shaping and modulating immune system. And so many folks in this call who are maybe involved in oncology have seen the data that shows that different microbiome impacts on cancer outcomes, treatment outcomes, or even getting cancer, et cetera. The abundancy and diversity of soil microbiome has been threatened by modern lifestyle, but there's an opportunity to restore this situation by a deeper understanding of soil microbiome. So, most people that know anything about soil know that it's a complete complex system, and that the soil is where a lot of our food comes from, and disrupting that system does have negative effects. And then microbiome animal husbandry. we were talking before this started about methane emission from cattle, trying to work with the microbiome in a way where we reduce negative effects that come out of animal husbandry and that industry. Here's just a couple more, won't take up too much more time, but perturbations in the gut microbiome favor the development of certain GI disorders like Crohn's disease, ulcerative colitis, ulcers, stomach ulcers, etc. So, and some of these discoveries over the years have really helped people deal with those debilitating diseases. Alterations in skin microbiome contribute to dermatological conditions like psoriasis, dermatitis, acne. Impairments in microbiome function may also dictate the metastatic fate of some cancers. This is what we were just talking about before. Human microbiome contributes to the shape, the success of cancer immunotherapy. Machine learning approaches based on the microbiome data can accurately predict glycemic responses to food intake. This is going to even increase. We had a meeting this morning with somebody who was just talking about this and how the microbiome and other ancillary factors will improve quality of life for diabetics and other endo-type situations. Human populations more exposed to interactions with soil and performing traditional agricultural practices are less susceptible to autoimmune diseases such as asthma. Rumen microbiome composition in cattle dictates the levels of methane emission. So there's so much work to be done, and it's comprehensive in our lives around us. So we have cause and effects that I talked about, you know, where you live, how that affects, impacts your life, agricultural, antibiotics, red meat in diet, presence in fiber in diet. All these things are leading to, in some cases, an enhanced quality of life, but in many times an unknown lower quality of life. And so the opportunity is there to make a big difference from an effect perspective. So as we talk about this, there's a really important lesson that we've all learned in the past with dealing with scientific data. And that is we have an opportunity here from microbiome research to do it right the first time. So adoption drives rigor, reproducibility, and model quality data. These are big lessons learned in large R&D organizations. Scientific data and process strategies, really writing down how we're going to handle and manage this solves a lot of problems for a lot of companies. Adopting data standards. This is another critical, not only data standards, metadata standards, making sure that the data is contextually rich. Entity registration, uniquely identifying the things that you're working with so that as you collaborate with others, there's no mistake or no misunderstanding on what they're using or dealing with. An electronic laboratory notebook. A critical part in science today is actually capturing the scientific method in electronic format. This is very important. This is letting people know not only what you did, how you did it, but it becomes shareable and it spreads the knowledge for your colleagues. And then a unified platform ecosystem for microbiome research. This is where adopting a software, hardware type environment will actually allow you to drive the outcomes and your discoveries and innovation in a much higher rate. The reason for all this is complexity, diversity, and volume. Massive systems-based science, massive data diversity, massive amounts of data, omics data, imaging data, analytical and characterization data. It's overwhelming. It's like, you know, if you talk to folks that did genomics and stuff, the amount of data that will be generated over the next 20 years is exponentially higher than even what we're dealing with today, and we're struggling with that. So an example of what that enterprise informatics and IT environment might look like, it's going to be a multitude of systems, but having an environment that can manage your microbiome data, your multi-omics data is incredibly important. And there's all types of processes that go on around this. The highest level processes are request, sample management, test management, experiment management, analysis management, and reporting management. And these are the types of tools, along with your microbiome type platform, that will allow you to run an enterprise play in an R&D space. All right, that's it for overview. And I'm going to stop sharing. And we're going to, so first of all, I'll ask the whole panel, was there anything in there that you disagree with or that you find, you know, very encouraging, etc.?
Well, John, it's Anthony. I think you outlined the challenge, and it's a significant challenge that we're facing. But I perhaps just wanted to say that I really appreciated your introduction to the subject. But the topic, of course, today is unlocking the biorevolution as well as the microbiome. So I think I just want to say a few things about that. I mean, first of all, you know, when we think about the microbiome, you've talked about the soil, but it does, in the narrow sense, you know, we describe it as that bioreactor in the gut that translates what you eat into what you need to subsist. But it, in the broadest sense, represents perhaps 50% of the biomass on Earth. And we've co-evolved with these microorganisms, or from them, over eons. And we've systematically ignored or been challenged about trying to understand them. It's become topical more recently. But I think we need to focus on the microbe as a fundament of life and incorporate it into all our models of understanding in biology. And that represents a big challenge. But to the point about the topic today, which is unlocking the biorevolution, we think of the microbe, and we're not alone in this because we're listening to McKinsey and Company with their fantastic report on the biorevolution, Boston Consulting Group, Henderson Institute, describing nature co-design as opportunities to unlock trillions of dollars of economic opportunity over the next 10 to 20 years, but with the microbe as the unit of currency. So understanding the microbe, how it functions, the microbe in an ecology functioning together with other components, and the microbiome or the ecology of those microbes interacting with a host, whatever that host will be, is the challenge I think we're facing. And if we are successful in advancing the art and the science to address that challenge, then we will, I think, transform the economic opportunities, and there will be a phase shift in the economics that we focus on as a planetary system, as economy is going forward. So I just wanted to describe, to my mind, what is that even more profound big picture.
Right. Okay. Very good. Mike, what are your thoughts as somebody, as a leader in the world who is all over the gut microbiome and working with therapeutic clients, etc.?
Well, I mean, you've touched upon it. It is remarkable that the gut microbiome has co-evolved with us before we existed as humans, and that now we can trace mechanisms that go from gut secreted metabolites from bugs into the brain, and that in fact neurotransmitters in some cases are more abundantly generated by the gut bacteria than our own brain, that we are integrated in this way with this new organ, but the new organ is incredibly complex, as you said. It's potentially plastic, and that worries people. How plastic is it? But it's very, very complex. And although there's a data management issue, there is actually a knowledge capture issue, which we focus in a lot, which is that you can sequence what there is there, but you've got to get down in there, isolate the bacteria, they're mainly bacteria, but everything else as well, the phage, etc., and start studying them as individuals. And this reminds me quite a bit of the systems biology story a few years back in target discovery in the pharma industry, which is, can you model all the pathways and get the quantitative flows so you can design and model where the points are that you should target with therapeutics? And I think what happened is that it wasn't done, because although it was an informatics issue, there was a bigger issue, which is actually, can you collect the data? And so we focus a lot on collecting information about the microbes. And it starts with actually even being able to recognize them, because even purely from sequencing approaches to recognize the bacteria in the gut, and that's one niche we're talking about, but there's many other niches we're talking about in this panel. is if you go with the classic shotgun metagenomics, we find you cannot classify half the sequence reads, public sequence data. So then we go in and isolate the remaining bacteria, get the reference genomes in, and then we can classify everything. So there is an enormous gap, even in classifying the most basic data here. Yeah. And that's before we have the issues of managing the data, bringing in the machine learning. And there's another aspect. You have variation between patients, and that requires sufficiently large clinical data sets to get the stats on the individual humans. I won't go on, but that's okay. That's just an opener, really, on the issues as I see them. You've got patient diversity. You need big enough groups. You've got being able to see the bacteria even, and then pull them out, culture them, and analyze them. And then you pull it all together, and you can get predictive biomarkers and drugs. But there's a lot of actual biology along with informatics.
Right. Okay, Barry. I know you know. You're focusing on some different areas, so it'd be really important to hear your feedback on some of this.
Yeah, I think it's really interesting. Anthony and Mike have actually already done a very good job of highlighting the complexity of what we are trying to do here as a community, as a microbiome community. I think my first interest in the microbiome was it literally started from my very first lecture in university. The very first lecture I attended was called "Microbes Are Everywhere." And nothing has changed. Microbes literally are everywhere. They interact with us on a daily, weekly, monthly basis, and they control so much of what we do on a day-to-day basis. So as Mike has already outlined, the microbes in the gut are so important in terms of overall body health. And I'm sure we'll get into much more information about how the gut microbiome impacts health. But I think from a Unilever point of view, the microbiome that we are interested in, as well as the gut, is potentially the cosmetic microbiome, the microbiome that lives on our scalp, the microbiome that lives in our skin, the microbiome that lives in our oral cavities. And the consequences of overgrowth or dysbiosis in those microbial communities is so important to understand. And I think a lot of the consumers out there at the moment, or a lot of the general public, have heard about the gut microbiome, and they understand that the gut microbiome is something to be studied and to be understood. But even recently, when I spoke to a group of MSc students in KCL as part of the Masters in Microbiome Science down there, I asked a simple question in terms of, well, how many of you have performed a microbiome experiment? And there was maybe one or two people in the class who recognized this as a question of, oh, how many times have I gone into the lab, I've grown up microbes, and I've actually studied these microbes. None of them were able to make the connection to really that question is, how many times have you washed your hair, washed your hands, brushed your teeth? Because in reality, they are all microbiome experiments that each of us carry out every single day. And I think once more and more people understand that our daily actions impact the microbes that live on or in us, so it really shows the complexity of what we're trying to deal with.
Yeah, absolutely. Dana, for somebody that's actually a microbiome scientist, hands-on in the lab, how do you take some of this? Where did you see some of your challenges? Is it a lot harder at the bench than a lot of people know, et cetera?
Possibly. I mean, having, experience in the wet bench and experimental side of things, as well as the computational, designing the experiment, first of all, that's a challenge, because for mouse models, for example, human and mouse microbiota can be so different. So you have to take that into effect. You can't, directly translate those results. But then there are other things you have to think about in a mouse model, such as they're coprophagic. They're replicates in the cage. So you got to take into account, well, actually all my mice in one cage are just technical replicates. They aren't an N of 3, for example, for your 3 mice. And then carrying that through all the way to the data output, then you have to think about, okay, well, I'm getting these huge data sets. Now, how do I analyze this? So it's really important to have your pipelines and your plan in place first, and then be familiar with all the statistical methods out there, because the statistics is also a field that's constantly changing and evolving, and there's new thoughts on how to appropriately analyze the data, and there are new methods out there all the time. So it can be a challenge to sort through all of that and decide, well, what's the appropriate approach for my data? So all things that are important to take into account when planning experiments. Right. How do I get meaning out of my data?
Yep. And Arlene, head of innovation, how do you how do you even wrap your head around all this, and how do you get started, or explain some of the what you've seen?
Yeah, so many great comments from the panel today. One thing that we really think about that I haven't heard mentioned yet is we also think about how this can apply to animal health, and how kind of the same things that happen in your gut or my gut are happening in a chicken's gut or in a cow's gut. And I like, John, that you mentioned methane reduction. The same goes for nutrient digestibility and digestion, right? And how that change in that microbiome can impact both producer profitability in animal health and do things like reduce antibiotics. So I think that is also a great point to call out. From a standpoint of how we think about this in a discovery or an innovation perspective, I think the most important thing is the data, right? And having that information to be able to call what is a good and what is a bad microbiome. And that is definitely a controversial question, I know. But for us, foundational is how do you get the samples and the data to be able to start to move down that path.
Got it. So then I go to Jeanette, because this is a perfect lead in for Jeanette. And, this is part of your responsibility of these environments, data environments, and making some understanding out of all of this. What's your challenge, and where do you see some of this going based on some of the things that you heard today?
Yeah, so I sort of go back to the comment Mike made at the start. At the start of this, I don't know, you can go back probably 15, 20 years, and everyone's like, there's all this data, and we're going to be able to get all these insights. And it always takes 15 to 20 years to get enough to be able to start pulling all the pictures together. And, people say there is a lot of data, there is also a lot of gaps. So, what we've been trying to do is it's about consistency. I think everybody's mentioned this, consistency of your data capture, consistency of, of the way you analyze that. So you're analyzing data in really consistent ways. And then really, I think there's the piece where the in silico part really adds to this is, how do you fill in those gaps? You'll never have every single bit of data. So how do you do the network biology, and how do you make those inferences so you really get the knowledge and the insights from the data that you've got? And it is a lot. And so you've got that complexity there. So it's really about the pipelines and the networks that really get you the knowledge. And so that's where we've been working quite a lot over the last few years. And I think now is the time when we have enough data. There's still gaps, but it can start to really turn a corner in terms of putting some of those networks together. So that's what excites me at the moment. It's also another big challenge, but I think that's the sort of capability that you need to build up as we go through this journey as a community. Okay,
awesome. So from a sustainability perspective of the biorevolution, what does that mean to Eagle Genomics? What, Anthony or Alex, from us, what does that mean to you?
Well, I'll start perhaps, John. I think we think about the grand challenges of our age. Perhaps there are only 60 harvests left in the soil. According to some commentators, that's the case. Why? Because we've destroyed perhaps 40% of the biodiversity in the productive soils. And unless we address that challenge, we're going to compromise our ability to grow foods. Because of compromised microbiome in the soil, soil plant interactions are compromised. So the plants we're growing are compromised in terms of the nutritional content and nutritional value available to the organisms that eat those plants, animals and humans. We're seeing over 70 years the breakdown in what I'd describe as the conversation between the microbiome and the host, because of the collapse of diversity within the host, which is causing all sorts of challenges. And we have unsustainable consumer value chains to try and get calories to consumers, for instance, which we need to re-examine based on, not anymore just delivering calories to humans, but health benefits to humans and indeed, as Arlene says, to animals. But if you flip all of those grand challenges on the head, those are massive scale opportunities. So we focus on trying to address the differences and the commonalities in addressing across these industry domains the data challenges. And I'd say, also there are a number of significant elements we need to consider when we're thinking about how to address the challenge. I mean, as Arlene says, that the $64,000 question is, what is a healthy microbiome? Well, we need to design experiments and conduct experiments to generate data and evaluate those data in ways that help us to answer that question. Perhaps we're looking at the wrong things. If we're just looking at genomic content of a microbiome, even if it's down to the strain level, is that the right question to be asking to understand what a healthy microbiome is? If we look at metabolism is largely conserved across the geosphere, the aquasphere, the hydrosphere, the atmosphere, and the biosphere. There are common metabolic pathways across rocks and humans, for instance. And when we think about the microbiome, there's a metaphor that has started to be used recently, which is that it's the song, not the singer. The song continues to be sung, irrespective of who's singing it. The microbiome can tolerate a degree of collapse because of the functional redundancy, up to a point. Beyond that point, you no longer have organisms supplanting each other to perform their part of the metabolic, metabolism. So we need to design experiments to help us address, the challenges, so that we can focus on the transformational impact in addressing these grand challenges, I think.
So just to come in on that, I think that's going to require an evolution in thinking and an evolution in experimental design. Have we got the right data points? Are we looking at the right things? Have we got long enough time series? Are we catching frequently enough? Have we got enough samples? And if we're going to move into network science, which is where I think we are going, because that gives us all kinds of novel advantages and ways in which we can look at shifting networks, which is really what we want to do, then I think we're going to have to evolve both the experimental design, but also the analysis side of things.
If I may chip in, we're deeply involved in this kind of work, and also I'm aware of different levels at which these questions apply in the therapeutic human arena. You've got the gross dysbiosis that you get after C. difficile and the fact that you want to replace the whole microbiome, but that also happens after treatment with cytotoxics, for example, in GVHD, and you want to replace with reconstitute the microbiome. So you've then got more major reconstitution. But if you look at, for example, the response to checkpoint inhibitors in cancer, they're not healthy or unhealthy microbiomes. They're all healthy microbiomes, and they don't look very different at the gross level. You really have to look carefully to find differences. We've found differences, but it's required sufficiently large patient groups, really careful collection of the samples, because of course they die in air, and a lot of reference genomes to be able to see the bacteria with such precision that you find signatures within that complexity. So I think there are different classes of questions here. And then, you also have if you talk about sustainability, something on the health side related to the agriculture side is that there's a lot of interest, for example, from groups like Gates in malnutrition in the microbiome, and the fact that stunting may be related to the microbiome and could be corrected. That's probably more gross. But then if you look at vaccine response and COVID-19 resistance, there's already indications in both of those, and vaccines with flu a few years gone by, that immunosenescence, as you get older, there are changes to your microbiome, that your immunological robustness is different, and that we may be able to correct that. That may be more subtle. So it's difficult to generalize with this, but you do need good data sets to begin with, because as Anthony said, there's degeneracy, and different bacteria could perform the same function And it may be that we don't get to that until we start forgetting about the whole genome or the whole taxon, but looking at the gene level. And everyone's moving towards doing that as well at the moment. So you may have signatures of bacteria which are redundant and they cover the population. But when you look at the gene level, maybe you'll get a core of a few genes which cover that genomic and biochemical correlation without needing so many different bacteria. So I think these are all interesting directions that will go, but a big drawback, a big barrier in the human side is lack of people collecting good clinical data sets in big enough populations. The pharma companies should all be collecting stool anaerobically from their clinical studies because it'll be affecting their drug response. Still difficult to get those samples.
That's a great point.
I'd fully agree with you there, Mike, and I'm all on board in terms of collecting as many samples as we can from as many different patients as we can across as many different conditions as we can. And I'd extend that out to things for Arlene, like soil samples. We should know what the microbiome of the soil is all over the planet. There needs to be initiatives to look at these things. I think, however, on the sustainability front, and I'm going to go a little bit broader here than just some of the things that Unilever are doing, particularly in the areas of plastic and renewables, et cetera. But to broaden it out in terms of the microbiome space, I think there are so many really, really exciting opportunities where the microbiome as a whole, or individual microbes, can actually help us solve a lot of the grand challenges that Anthony talked about already. I'll defer to Arlene in terms of the soil microbiome. I'd really love to hear her thoughts on how, for example, how the microbiome can alter the nutritional profile of plants. So I'll teeing you up, Arlene. I'd really love to hear your thoughts on that one. But even some of the work that's going on already, as Mike said, in terms of looking at genes and looking at enzymes in microbes that can, for example, digest plastics. Well, that's an obvious place that we need to be looking for microbial enzymes that can essentially do chemistry that people can't. Because whether we like it or not, microbes are much, much better chemists than we will ever be. I can say that as a biologist, and I know that sometimes annoys some of my chemistry friends, but I truly believe that the microbes are amazing chemists. When you look at some of the genetic engineering that is done, I believe there was a paper a couple of years ago from the Weizmann Institute where they've engineered E. coli to ingest and utilize carbon dioxide as an energy source. It's absolutely fantastic. These are some of the things we can do. We need to be mindful of the fact that whereas everybody thinks of the Amazon as the lungs of the world, and don't get me wrong, it is absolutely essential. We need to remember, however, that cyanobacteria produce 20% of the global oxygen. So the importance of microbes cannot be understated in terms of what they could potentially do for us in these sustainability challenges.
I think you're pointing out a lot of biases that have got us to where we're at today, from the first, observations of the microbiome, that we tend to bias a lot of this over time for a multitude of reasons. And this could be a whole nother segment of talking about that. But the point that you just brought up, and you hear it all the time, ooh, save the rainforest, of course, save the rainforest, but everyone forgets about where they live, right? So that brings it home and says, yes, we do need to take care of where we live. And one of the ways of doing that is investing in what everyone just talked about here.
I think before I pass over to Arlene, just one more. There's a quotation I read in a paper recently, and I think it was mentioned by Tom Curtis at the University of Newcastle. And I'm paraphrasing, but he said something along the lines of, if the last blue whale killed the last great panda, it would be a tragedy. But if the last methanogen killed the last ammonia oxidizing bacteria, we'd be in much, much bigger trouble. And I think it's a really nice way of thinking about how important these microbes are.
All right, Arlene.
Yes, and Barry, I wish that agricultural microbiomes were more my specialty, but I think you call out a great idea that there are a lot of underexplored areas besides the gut microbiome, right? We see so much research and so many great outcomes happening as a result of the gut microbiome, but there is so much potential in this other additional space to, let's say, carbon sequestration through plant, changing what the plant's chemistry is, things like that. We've looked at spaces like milk microbiome, or skin microbiome as well. And I think it tees up actually one of the big challenges we've found in the space, and I would love to hear some thoughts from the rest of the panel on this, is some of these low density applications. So easy to look at, easy in quotes, to look at a microbiome in the gut or in other high density things where you have a low host cell population. But in some of these other spaces, whether that is plant surfaces or skin applications or other applications, it becomes really challenging to kind of understand that background post DNA, and being limited to some of those 16S technologies really limits the richness of what one can do in that space. And so I would personally be curious for some thoughts from this team too on, you know, some of the limitations they see with that science, and how we can help grow and progress in that space so that we can go into some of these additional applications outside the gut, outside the oral, outside the high density microbe populations.
Aline, I was amazed a few years ago. I interacted with a colleague who was involved with a plant microbiome company in the US. And I believe he told me that microbes were already used to control infection in crops instead of pesticides, I guess. Is that correct?
They are, especially in organic agriculture. There is, oh, looks like Mike broke up for a second. Particularly in organic agriculture, there is a lot of biological control. There is actually phage used commonly in plant agriculture as an alternative to chemical pesticides as well. So there is already people starting to leverage this, particularly in those natural organic solutions.
Yeah, it was my first internship, Mike, was Bacillus thuringiensis was used. It would produce a toxin that would kill potato bugs, right? So that was one example that I'm very familiar with.
Hmm.
So Eileen, I'm going to let Barry talk about kind of low diversity environments like skin, because he's the expert in this. But I just would say that, you know, a lot of these haven't received the same amount of attention and detail as the gut microbiome. So for a lot of these things, we don't even know what's there. You know, 3 years ago, we didn't really know what we knew and what we didn't know in the gut. There's been a lot of attention, a lot of bioinformatics, a lot of culturing has gone on. And I think we now have a much better handle on that. But for some environments, we just don't even know. And we're only really starting to get to that sort of level of knowledge. So, but I'll hand over to Barry.
Before Barry comes in, I'll just say that I am aware of it because I've seen a lot of investment, a lot of pharma interest in skin microbiome. You know, GSK did a deal in, I think, a phage to target one of the bacteria involved, but they're not the only ones. And actually my investor, Saventure, is involved in the microbiome across food and skin and cosmetics as well. So I think it's a very exciting area.
Yeah, I agree, Mike. I think the deal you are referencing, I believe it was GSK and Eligo Bioscience. I think they're looking at bacteriophage for C. acnes for medicinal applications. I think the numbers suggested are in the $150, $200 million range. So it's definitely a big deal and extremely interesting. I think Alex's point, however, is well made. And for the likes of myself and some of the team in Unilever, where we look at the skin microbiome a lot, we look very jealously over the fence at the people who are doing the work in the gut microbiome area. I think a huge amount of the methodologies, the sampling, the informatics, the analysis is led by the information in the gut microbiome space. But I'm very jealous of the fact that the idea of metabolomics, metaproteomics, metatranscriptomics are things that can be done with the gut microbiome. As far as I'm aware, and if somebody on the chat can point me in the right direction, I'm not aware of anybody doing metatranscriptomics analysis on the skin microbiome. I think is a really key gap that we have, to look at the gene expression of the microbes. What are the microbes doing? I think we're getting there in terms of the what is there, but we still have an open question in terms of what the microbes are doing, and it's really getting to the functionality that we need to. But yeah, I think that's only the part of it. But Mike, I know you've done a huge amount as well in the culturing space, in the gut microbiome. So if you're ever interested in maybe looking at the culturome of the skin.
My scientific founder, Trevor Lawley, has actually explored the microbiome in other areas. The bladder was thought to be sterile, and he published the whole microbiome. He cultured all of them, the bugs in the bladder. He's done it in other areas. He's looking at the lung at the moment. So he's got a bit of a mission to go to different areas. The skin, I mean, you know, there are issues with the gut, of course. And I don't know if I mentioned it, but one of them is it's difficult to routinely know about the differences in biology in the small intestine versus the bowel. And then it's quite difficult to look at bacteria which are in the mucus layer or very close to the epithelium, because, as I think Arlene mentioned in passing, you can't do metagenomics. You have to do 16S, because if you do metagenomic sequencing, you capture all the host DNA, which massively contaminates it. So you have to use a lower resolution technique. So I think there is scope even in the gut. I think we know about a lot of bacteria, but there's a lot we don't know about how they interact with each other, where they're located exactly, and what they're doing in different parts of the gut. If we had something spatial, that would be really good, but there's a long way to go.
There's a quick question there, Mike, around the diversity of the gut from a microbiome perspective. Is it large or is it not large? I won't even pretend to answer.
Well, let me answer it this way. The first thing is that, I'm trying to work out where to start it. A few years ago, there was a publication from Rob Finn at the EBI with Trevor Lawley at Sanger Institute, and they looked at metagenomics, and they estimated that there were upwards of 10,000 species of gut bacteria. And by that, I mean healthy gut bacteria, commensals, not pathogens, because you can get a lot of pathogen species. And the thing is, there's much more recognition of the pathogens. They've been categorized, they've been cultured, there's loads of strains. But on the healthy gut commensals, only a minority had been isolated, maybe 10%, maybe now it's 20%. And that is an enormous gap. They've been viewed as unculturable. So this metagenomic approach estimated that there were upwards of 10,000 when there were fewer than 1,000 isolated with reference genomes. And that's a big gap. Now, when the same species, any one human has upwards of 200 species in their gut. We can now culture them all from one person. We can culture everything from one person. Upwards of 200 species, but there are many strains. And if you have the same species in another person, it will be a different strain. They change, and so there are strain variants. And because the biology varies by strain, you have to get to the right strain from the human where you saw that bug do something. You can't just say, we found something, we think it's this species, or even worse, we think it's this genus if you do 16S, and we'll try out what this bug does in the lab. You've got to go to the same one, ideally the same strain. So there are upwards of 10,000 species, and there are millions of strains. But each of us has maybe 250 species and loads, hundreds of thousands of strains. And that's just the bacteria. Of course, there are other things in there.
Very interesting. Let's see. You know what? I don't want to even hog this as a moderator. Is there anyone on the panel? There's a lot of questions out here. There's a lot of, here's an anonymous one, but you guys also think of a question to ask the panel as well, because I think it would probably be better than one I would ask. If the primary benefits of good microbiome health end up coming from things like better, more scientifically informed hygienic behavior, better dietary practices, and the like, and not drugs or proprietary products, where are the profits opportunities going to come from necessary to pay for the complex scientific research? So there's this conundrum, I think, where the question's being asked who's going to pay to investigate really what is the next frontier for us? It might not even be Mars or other planets. It's actually, we haven't even characterized our own planet yet, right? Where's this all going to come from? I'll just open it up to anybody. Go ahead, Anthony.
I can provide a high-level
Okay, go ahead, Arlene. You go first.
Arlene, you go first. Yeah, please.
Yeah, I think as terrific as it would be for everyone to change their diet and control their environment, unfortunately, it's sort of unable, or people are unable, unwilling sometimes to make those changes. So I think there will be a place for supplements and therapeutics to help, either readjust and get you on the right path when you had to take antibiotics, and you might have dysbiosis that was created from that. Or, if for some reason you have dietary constraints and you can't kind of follow a standard diet protocol. So whether that's one of those two paths, I definitely think there's a place for products. And I do think some of that investment will drive forward the science.
Okay, Anthony?
Well, I think, there's a sort of industry transformational dimension to answering the question you posed there, John. But I think there's opportunity perhaps over the next 5 to 10 years for the perfect storm, because we're seeing huge dynamics in the alternative protein space, transforming what we eat. And as Barry said, in a conversation in the green room prior to the call, sometimes it's not easy to differentiate between an alternative protein burger and a traditional burger. But we look at the technologies being applied to creating alternative proteins, first plant-based, then biomass fermentation, and then in the next generation, precision fermentation, and then of course, perhaps cellular meats. The technologies that are going to be required to develop these alternative proteins are going to rely heavily on an understanding of the microbe. So precision fermentation, I talk to R&D leads of precision fermentation companies on a regular basis. At the moment, sometimes they're scooping up handfuls of earth, conducting sequencing analysis to understand what's in there, whether it's utilizable or not. So the gut is a bioreactor. These are, bioreactors we're creating, and we're trying to put together strains so that we can target a particular precision fermentation outcome. We've only sequenced 1% of the soil. So the challenge is as big there as anywhere else. But I think there's a huge investment from the VC community in alternative proteins, which is going to drive the art and the science. There is a fundamental requirement for us to address the challenges of antibiotic resistance. And Mike and his company in building their therapeutics propositions are seeking to go at that. We have to solve that challenge. And we are talking to and working with World Business Council for Sustainable Development, FRESH initiative, looking at fundamental re-architecting, the food industry, and the EIT food and health groups, who are all trying to, in preparation, by the way, for the UN Food Summit later this year, if it goes ahead, catalyze the community of industry and the scientific community into trying to re-architect, how we produce food, how we eat, to address some of these grand challenges. So I see across industries investment and sort of behavior to catalyze transformation, which I think is going to yield slow but inexorable shift over the coming years.
Yeah, if I may, I think, of course, there is a lot happening in academia too, and it's sometimes some of the technologies aren't available, but in some cases there are big technologies available. The Sanger Institute is one case. The Broad is another place where there's a lot going on. And there's more and more happening in China. People are beginning to get culture collections, doing the genomics and doing the other biology, metabolomics. And then you have a whole slew of different companies in different sectors. In the health side, you have, you have probiotics as well, which will be lower cost, but which will have an impact. There's one application I think is sort of in the crossover. And I may mention a company, Evolve Biosystems. I admire what they're doing, is they're trying to take the state of newborn infant gut microbiome back to what it was before we started messing around with our lifestyles, because it used to be monoculture. I mean, there's lots of evidence that it used to be monoculture. There used to be bugs which were good at protecting from pathogens and teeing up the immune system that colonized infants as soon as they were born. And then gradually they receded. But now the early life stuff shows that it's not as much monoculture, and there's lots of antibiotic resistance and pathogens from hospital births, etc. So in this particular case, they're looking at a probiotic approach to reset back to normal. Now that could be applicable widely, could be low cost. So there could be solutions like that in health which are, you don't have to know everything. We've talked about the complexity, but let's not get too depressed about that. In the therapeutic space, there are consortia of products that I think will come through in the next few years. We've already had successes, there'll be more. Without knowing everything, we'll get therapeutic applications. In that kind of area I just mentioned, I think that'll work. And so I think as we learn more, there will also be successful applications along the way. So.
Oh, absolutely, absolutely. I mean, if you're an entrepreneur and you're on this call, your head should be spinning. Well, that's all I'll say about that. Here's a question for Eagle Genomics. So Alex and Anthony, from a design of experiment perspective, when researching the impact of various interventions on human health, how do you go about controlling enough of the exogenous conditions to sort out signal from noise and results? So this is a broad question, but I mean, you might be able to exemplify parts of your platform or whatever that maybe could help with this.
Well, I mean, that's a very big question. That's where we bring in the kind of data science team, or we get the involvement of statisticians. I mean, the important thing is trying to work out what question you want to answer, right, before you start your experiment. And a lot of experiments, and I come from originally an academic source, was you would have certain funding, you would work out how much sequencing you could do based on that funding, and then you perform your experiment, which isn't really the right way to build these experiments up. What you need to ask is, what is the question I want to answer? And then is the amount of the number of samples I'm going to get, are the number of measurements I'm going to get, are they actually going to answer that question? Are they going to give me the statistical power? So I don't know if you want to add anything else on that, Anthony.
Thanks, Alex. I would say, we are going to have to address some of the challenge of how to design experiments to generate results that are reliable as we move into sort of generation 2 of our understanding of the microbiome. I've seen some fabulous papers, sponsored by the Royal Society in the UK, asking for us to design hypothesis-led experiments rather than, because what we're seeing is the field advance significantly on the basis of new technologies available. Technologies are being exploited. We're getting results from, analyzing samples with these new technologies, developing sort of descriptions of what we see, and then backward engineering hypotheses. And that's good, but it's not sufficient. We have to focus more on hypothesis-driven science as well in the next phase. And the other thing is, when we think about the approach to science, evidence-based science, where we design experiments such that we have a reliable, statistically reliable result at the end. We can be confident about the reliability of the result, but it doesn't necessarily tell us what we need to know about, the impact of the result, because we're not taking into account the ecology effect. And that's a very, very significant dimension of the challenge we're addressing here. And so that's why we, in applying network science to trying to address the microbiome challenge, are seeking to provide that ecology dimension. And I think that's a very important thing. There's been this exponential increase in activity and scientific discovery over the last few years, at least. We want to be able to network the science that's been conducted there, but also build a reliable foundation that we can continually augment with further understanding. And once again, I think the application of network science to that challenge is to our minds one of the most reliable and exciting approaches.
So an applicable follow-on to that then is the data fabric question here that says, is a data fabric, a woven data fabric, enough to capture the microbial interactions, et cetera? And I would suspect over time, but what do you guys, how are you guys viewing this from a data fabric perspective?
Well, I'd say that we, believe that just for scientific endeavor and for the acceleration into the biorevolution, fundamentally, we are going to have to build reliable, robust data fabrics, so that we can explore these data and generate results in a meaningful, reliable, repeatable way. And the exciting thing is a whole slew of enterprises that we're working with acknowledge and recognize that. So, and I think that's the beginning of it. But we also need to find mechanisms to enable scientists to collaborate around these data, humans to interact and converse around the data and with the data. So a data fabric is necessary, but not sufficient. A data fabric that unifies and links the data within an enterprise, but also networks those research establishments, whereas Mike says, is, you know, the art and the science is really being driven, so that data are available to enable collaboration between research institute and enterprise. But then we are focusing on building a reliable application framework on top of that data fabric, so that we can enable this, you know, systematized interaction in a reliable way, so that we can support the scientific endeavor from, you know, the most upstream challenging part, which is hypothesis elicitation, trying to combine entities of interest that might form the basis of a hypothesis, filter those hypotheses, conduct exploratory data analysis, generate results, and then provide evidential material to support a microbiome-based product claim, for instance. That application layer is necessary as well. So to our minds, the combination of the data fabric and the application layer is going to help us advance more quickly.
Okay, I have a question. Go ahead.
No, I just want to, so building on a lot of what was just said, I think, you know, there's another layer, which is the people and getting the right people in front of the data. So you've got the data level, which is about consistency. And then there's the hypothesis led. And I think we've moved away a lot in the last year from just saying, here's all the data, you know, it's efficient at what can you find, to we know what the knowledge base is. How can that data integration and data system help us ask questions and come up with maybe several hypotheses, which we can then go and test. But I think the other aspect of it, which has developed a lot over the last sort of 5 years, is who is doing that, who is looking for those insights. So, you know, in the past, we might have said, well, here's all the data, we'll hand it over to the data group and they do all that. And then they pull it back and send it across with the results. And I think more and more, you know, the people with the biological knowledge and the people that you want to have the lens looking at that data in a distilled way are the biological community. And so I think, yeah, the evolution of tools that democratize and enable people to come up with analysis themselves, as well as getting the right people in front of that data, not just the data scientist group, is a big change, and we need to drive that forwards to do that. I really
recognize what you say, Jeanette, and you know, especially when you get into techniques like machine learning, which are a bit less transparent, and the biologists do need to be there to understand and guide or contribute to the guidance of what's going on. And really what you want is bioinformaticians who are very biological and biologists who are pretty informatic to act as the bridge, that they can talk to each other.
Absolutely.
But you've got a cultural issue there, you're right. And you'd need those bridge people.
And I think it's partly about, you know, it's always about lowering that barrier, isn't it, to that interaction, because you want all the sort of boring things, the boring bits of the analysis to be done automatically, because you don't necessarily need somebody sat at a desk doing all that. But you want the bit where the knowledge is coming together and somebody's making a decision, a scientific decision, to be in front of them easily. So, and I think we're at the point where those sort of tools are starting to come online and they're available. But, you know, there's a lot of work to be done there.
Absolutely.
If you look at those analysis chains, the data generation and analysis chains, they're long and they're complex, right? You've got very few places which are doing the experimental design, the sequencing, the analysis of the data, the machine learning, causal analysis at the end of it. There are a lot of different techniques along the way there and different specialties, and it's quite difficult to find individual departments even or companies which are doing all of that, all, you know, from one end to the other.
Yep. All right, let me get to audience question. This one is from Ross Youngs. He's the CEO of Microbiomics. They actually are able to take aquatic environments and sample the whole microbiome from every aquatic environment. His question is, what is the least common denominator in all microbiomes? Is it species, genes, metabolic pathways, or chemistry, or something else?
That's a good question. Oh, well, can I have a go at it, John?
Of course.
Well, I would say at the taxonomic level, it's undoubtedly strain. I mean, strain can be, as you know, in some people's eyes, it's SNPs difference. In other words, a few nucleotides difference from each other, and that can lead to different biology. So that, from a microbiological taxonomic way of looking at it, you've got to go to the strain level, because a species may have different strains with quite different phenotypic impact on the host. However, you could argue that if you break it down and look at it a different way, then it breaks down further. It's actually at the genome metabolite level. And, you know, it could be that a phenotype is due to one particular metabolite generated by completely different, not just strains, species, but genera or more, or even phyla. So then that's a different unit. So I think actually there isn't one answer. If you're being a straightforward microbiologist, you would say strain. But if you're looking beyond what individual strains do and look at degeneracy and redundancy across different organisms that do the same thing, then it's another thing. It could be a metabolite, but it could be where the function is coming from, whatever that is, you know.
Barry, you had your hand raised?
Yeah, I fully agree with Mike, but I think there is also an additional answer to the question in terms of it likely depends on the context that you're examining it. So, for example, the strain is definitely important as well, but at least from some of the work that we've done on the axillary microbiome, the underarm microbiome, it's all about what the consumer perceives. So in the area of the axillary microbiome, it's about odor. It's whether or not a consumer can smell odor coming from their armpit. And despite the fact that it's a topic that not many people cover, it's absolutely fascinating, the biology, the microbiology that goes on in someone's underarm, because again, it is about individual bacteria, it's about individual transporters, it's about individual enzymes that we've discovered that result in the generation of body odor. But there also is a human element to it as well. As I've just answered in one of the questions, believe it or not, there is actually a single SNP in the human genome that will determine whether or not you release a malodorous precursor, which is then broken down by an enzyme produced by the microbiome. So again, it's just lovely interplay between the microbes and the human host and how they look at it. So in terms of lowest common denominator, strains I'm all for, enzymes I'm all for, metabolites I'm all for. I think they all should be part of the equation.
That's pretty cool. Oh, well, you know, and that parlays into the food science, right? With cheese, isn't it? Different cheese flavors and things will come from the different microbiome associated with the head cheese masters and the people making the cheese, etc. So it's all fascinating.
Yeah.
Not only cheese flavor there, John. I mean, we know of companies that are seeking to reconstitute the diversity of cheese cultures, not just to tune for flavor, but to protect against invasive species, to protect against listeria incursions, for instance. So it's absolutely fascinating what we're seeing in the food space.
So there's a question.
I think Dana has a hand up in terms of the lowest common denominator from the lab scientist. Maybe one of the only one who gets to go into the lab anymore.
Sorry, Dana. Go ahead.
That's okay. No, I wanted to just jump in on this conversation because, you know, I agree the strain level is really important, but then, and we were talking about the metabolites, and I think this is where like the field is moving in the direction of multi-omics analysis and how do we integrate metatranscriptomics, proteomics, metabolomics in order to make more sense of that data, and maybe that would help us understand, you know, from an ecological perspective, what are these unique microbiomes doing, And, you know, from an individual medicine basis or from a therapeutic product basis, like how can we use that to predict, well, what's the outcome for this person or what product is going to benefit them?
Yeah, no, absolutely. I think that's the future. You know, given we're talking about all this, so when, you know, in the prelude, I brought up a lot of these things to say, hey, if we do these things, we'll be much better off down the road than if we all go at this individually and everybody goes and does their own thing and then tries to bring everything together, which doesn't work well. So a question was raised about data standards. Would, and I took out company names and all that stuff, and this is a question for everyone. Wouldn't it behoove us to all get together, corporations, .orgs, academics, and come up with microbiome data standards wherever possible to help this along? We're not saying it's the absolute answer. It's to help reduce complexity, help with the analysis, help with the decision making along. So I'll start with Arlene first, since you know, you're innovation director at Cargill.
Yeah, I mean, this is a little bit of a tricky one because I don't know that we're at a place yet where it's determined what that standard should be. Mm-hmm. So I would say from our perspective, we actually try to look at, you know, whether it's multiple models, multiple analysis techniques, and try to see if we can get to the same answer. Because we don't feel like right now there is one consistent, perfect method. I think we definitely support the industry moving towards that. But right now, I would say we haven't seen one particular standard model, method, technique that we feel like we can kind of put our support upon. So we actually look at multiple different tools and see if they converge to the same answer.
Okay. Jeanette, this is, you know, given how deep you are in data science and everything like that, is this something that you're seeing would be beneficial? Are you in the same spot as maybe Arlene and Cargill? Like what are your thoughts around data standards right now?
So, I mean, in terms of the data we collect, obviously we're as consistent as we possibly can be across the different areas. And we try to align that with the sort of external standards. In terms of the actual collection, in terms of a global position, I don't know, I'm not the experimentalist on this, so I might sort of pass that one back to the sort of experimental side to Barry. I don't know whether you've got any other thoughts, Barry. I know we standardize the data we collect in terms of everything.
Yeah. Yeah, we do what we can in terms of making sure that the analysis that we have is consistent. And when I say analysis, it goes back to how we take a sample, to how we make a library, to how we sequence it, to how it's processed. And I'll be honest, Eagle have been really helpful for us in that particular area, because we are in a position now where the many thousands of microbiome samples that we have processed now are sitting with Eagle Genomics, and they are all processed in exactly the same way, using the same pipelines, thanks to Alex and his team. And what it really means is that data we collected 5 years ago, for example, is just as valuable as data we collect today. The data we collect today, there might be a lot more of it because of the amount of data that you can produce from a sequencer. But I think at least having consistency in your own sample collection and your own data processing is really, really important. I think the key element that we're missing, that Arlene mentioned earlier, is that, well, how do all of the companies and how do all of the research academics all agree on a single set of standards? I still think we're quite a way away from that. Anthony?
Yeah, thanks, Barry. Thanks, by the way, thanks for the reference to Eagle Genomics, Barry. I think what we're seeing as the industry matures is the evolution of pockets where industries and industry alliances are seeking to focus on the challenge of standards. So particularly speaking to you and Bernie, who's chairman of the Global Alliance for Genomics Health, you know, they want to have a role to play in developing microbiome data standards and metadata standards. The NMDC in the US is a consortium of industry participants and research establishments also looking at the challenge. Most recently in France, we've seen the launch of Metagenopolis, they have a call out to focus on microbiome standards. And in the UK, we have the UKRI's Microbiome Innovation Network that recently published the microbiome strategy paper with a section devoted to how we can align with the emerging global standards. I think we're going to see a lot more of this going forward. I think we're also seeing, in relation to live biotherapeutic therapies, etc., the regulator evolving their position also on pre, pro, postbiotics and symbiotics. There's a regulatory challenge that's evolving in parallel with the standards challenge. And I think we're going to see a lot of movement in both of those domains in the next couple of years.
Yeah, very good. Mike?
I think, whenever this topic comes up,
I know it's one of your favorites.
I feel in two minds. Because firstly, we want to get hold of clinical data sets and make sure that they're standard and do something with them. And when I say that, I mean clinical trials that have been run, the stool has been collected carefully and aerobically so it's not deteriorated. And if there's sequencing, it's been extracted from, and the sequence has been done to a standard, and it's all been put together in a standard way. And we have our processes, and we've done that, and we believe they're top notch, like some other groups that are top notch, and that those groups can do it, and that there are many who can't. And then we have another level where we analyze what bacteria are in there, and that then depends on our reference genomes. And we will get a different result from other people because we have a much more extensive physical reference genome database. We believe it makes a difference, and that we can see other bacteria and see them precisely and quantitate them accurately. We don't really want to share that bit. So I think that there are companies for whom this encroaches into their competitive business model. But there are probably elements which don't. And we haven't been so active and so interested in finding those out because we're interested in getting the good data today. And we're not in the business of enabling lots of people to do that, like Eagle Genomics is as a business model. So I think that's just a consideration. I think for us, there would be an area where we wouldn't want to be involved in that, and there'd be another where we'd be very happy to see it happen.
Okay. Yeah, I think standards are a really important thing. I think I don't know if I invented this Conwayism or not, but I just have a rule of if you have consistent rules, you can consistently change. So these standards will evolve over time, but if you can at least get a basis and use those and over time evolve them, you'll be in really good shape. And our world has become very collaborative. So the easier it is to be able to talk with somebody at the data level with your data, whether metadata harmonization has taken place or all the things that you need to join your data together to make better decisions, will benefit everyone moving forward. There are so many questions on the chat and the Q&A. I hope you guys could go in and take a look at the Q&A and answer some of these questions. I'm going to touch on, Barry's answering one around phage, accelerating phage ability to control bacteria's response. So I'll let him take that. There's a question around AI ML companies out there for microbiome research. So has anybody worked with, outside of Eagle? Are you guys adding to your repertoire once you have, the analytics engine from an AI ML perspective? Do you want to touch on that, Alex? Or go ahead.
I've got a number of things to say on that. So I think so we're partnering with leading cloud businesses across the globe that are driving horizontal AI models that they're looking to apply in industry vertical applications. So we're developing relationships with companies like Microsoft so that we can exploit the AI and ML capabilities they're providing, incorporate them in our eData Scientist platform. But I think when we're looking at the challenge of biology, which we still don't know enough about, I think the traditional good old-fashioned AI approaches have got to evolve because they sort of rely, or at least symbolic AI relies on a correspondence architecture. We have all these axiomatic objects that we're seeking to manipulate that have relationships that describe how they interact. But if the objects that we're trying to describe are evolving and emerging on a constant basis, it's extremely difficult to create that correspondence architecture that makes the difference. Also, the deep learning approaches that don't have any concept of symbol are not sufficient because those are black boxes that we can't interpret. They're not white boxes. But I think what's really interesting here, particularly, is just in recent weeks, DeepMind, published a paper in Nature where they said we need to evolve our models. The standard AI models that we're applying at the moment, the two-party zero-sum game adversarial approaches, they're not going to help us evolve to address these grand challenges, like some of the things they're doing around protein folding. So they talk about more collaborative approaches that we need to engineer into our approaches to ML and AI, so that you can establish conversations between machines and humans and enable collaboration between humans supported by AI. I think we're going to see a huge wave of innovation there, and we're certainly focusing on that very hard at Eagle Genomics. Okay.
Well, look, it's already 26 past the hour. I told you this time would fly. I think I'll ask one last question here, and I'll make the questions available for folks if they want to answer afterwards, etc. But now that we have a whole nother understanding, or we're starting to understand the complexity of the microbiome, and many people are making very awesome efforts toward it, do you think that this will impact not only drugs that won't make it to the market in the future, herbicides, pesticides that will get pulled or would never be used? You know, you go down the line of, well, now that the microbiome is understood more and involved, will it actually make it more difficult to go to market with some of our approaches, etc.? Mike, you're thinking, I see the smoke coming out.
I'm very interested in the microbiome and drug response. I mean, as you know, I'm sure, there are quite a few examples of the gut microbiome affecting drug metabolism. So drug removal, you know, whether the drug persists, so it affects DMPK. So that's a crude, obvious thing that has to be taken note of. But, you know, if you take any immunological drug, the microbiome, the gut microbiome definitely has an impact on drug response. And actually it'll be the same in every area. So I believe ultimately, and the pharma companies are not doing it very widely, they've woken up in the IO area, because the gut microbiome is so obviously driving the response, and in some others like IBD. But it'll be many areas, and I don't think it'll be a negative that it will say, you know, fewer drugs will make it. I think it will be enhance our capability in the clinical stages for patient stratification, patient selection, and therefore ultimately coming together with the human genomics. The microbiome genomics is as important. There are papers to say it's as important to make for precision medicine, which means which patients do we take, which ones do we treat. The microbiome will be part of precision medicine.
I like your answer. Arlene, I'll ask you from a human health perspective. Maybe you're more on the side of herbicides, pesticides, other things that we see in nutrition and farming and all that stuff. Do you think this will impact it positively or negatively, or right down the middle?
I would say positively. I think we already see this from a perspective of where antibiotic use in feed has changed, as an example. So here in the United States in 2017, a veterinary feed directive to say you can no longer put subtherapeutic levels of antibiotics into feeds.
Right.
We've already seen that kind of pivot and increase the interest in the industry towards these other sorts of compounds, probiotics, prebiotics, postbiotics, that can positively influence animal gut health. And so to say you have a viable alternative to antibiotics in some of these situations that can enhance performance, improve health, I think it's really attractive, and it starts to become more and more, I think, pressure on the value chain and on producers to say, why are you using this anymore when there's a viable alternative?
Okay, awesome. And then Barry, I'll go to you.
Yeah, again, I'd link back to some of the things that Mike said in terms of the personalization element to it. If therapeutics is is what is needed, and the impact that the gut microbiome and other microbiomes can have on outcomes for particular things. So I've done it myself. I've no affiliation with the company, but I've recently sent off my Skin Trust Club skin microbiome sample collection kit. So I think we're definitely in an era, partially as a result of COVID, I guess, where people are now getting used to sampling at home. So if you can take a COVID sample at home, why can you not sample your skin microbiome, your oral microbiome, your gut microbiome? These are the ways in which we're going to generate more and more data and help consumers get the outcomes that they really, really want.
Fantastic. Well, Anthony, you want to get the last word in? Anything you want to say on that?
No, I think that the Barry's crescendo was fantastic.
Yeah.
I think we're starting to see some panelists suggesting that they need to jump onto other conversations with English and German participants.
Okay, I wanted to thank everyone. I thought it was awesome. We really got to crack open this a little bit and start talking about it. I know from the comments that we got, people really enjoyed hearing the diversity of everything that's going on, how things are being done, and everyone sharing their experiences and et cetera. So I thought it was awesome. So thank you very much for taking the time today for this.
Thank you all. Thanks, John.
Thank you very much.
Great panel.
Yep.
Super.
All right.
Thanks, John. Really
take care, everybody.
Pleasure.
Cheers all. Thanks,
Bye-bye.
everyone. Bye-bye.
Stay healthy. Bye.
Stay healthy.